DNA polymer or deoxyribonucleic acid is made of many monomers called nucleotides. They are the basic structures of the DNA strand. These nucleotides are further divided into three parts: the deoxyribose sugar, a phosphate group, and the nitrogenous base. The first part of the nucleotide, the phosphate, is a phosphorus atom surrounded by oxygen, a simple common structure. The next part, the sugar, is called a pentose sugar because the carbons form a vague shape of a pentagon. The carbons are labeled 1', 2', 3', 4', and 5', carbons that will be used later. The next part are the nitrogen bases. There are 4 different types in DNA named adenine, thymine, guanine, and cytosine. They are abbreviated to the letter that they start with: A, T, G, and C. They are separated into 2 groups, purines and pyrimidines. Purines have a nitrogenous structure of a hexagon adjacent to a pentagon, while pyrimidines only have the hexagonal structure. Together they make a DNA strand that is like a twisted ladder, a double helix. To hold this structure together, the monomers bond to each other. Between nucleotides on the same side, they form phosphodiester bonds that binds the 5' carbon phosphate group and 3' carbon of another nucleotide. In between the two sides of the ladder are hydrogen bonds that hold the nitrogen bases. The hydrogen bonds only form between pyrimidines and purines, so it could fit in between the DNA strand. Furthermore, C can only bond with G and A can only bond with T. This is because there are 3 places on guanine and cytosine to form hydrogen bonds and 2 places for adenine and thymine. Altogether, they form the structure of a DNA strand that houses all the information of a cell. The next topic is DNA replication. In the cell cycle, the DNA must replicate so that the daughter cells will have the same amount of DNA and can do all the vital functions the parent cell can, creating clones. In order to do this, it needs to go through a series of steps. The first step of DNA replication is when the origin of replication is found by helicase, an enzyme. The helicase then travels in between the sides of DNA, breaking all the hydrogen bonds in between the nucleotides. This forms a replication fork and splits the parental DNA into 2. Also, hydrogen bonds form spontaneously if unchecked, so to keep the hydrogen bonds from reforming, another enzyme, topoisomerase, physically holds the strands back. Once they are finished, a pair of RNA primase and DNA polymerase 3 travel to the origin of replication. Fun fact, there are actually 11 DNA polymerases, but that is a video for another day. Because polymerase 3 cannot attach nucleotides to hydrogen bonds of the parental DNA, it needs primase. DNA polymerase can only attach nucleotides to the 3' carbon side of the DNA molecule. So, the primase initiates the process by replicating a few nucleotides. This gives the go sign to the DNA polymerase. DNA polymerase 3 creates these nucleotides by using nucleoside triphosphates, which are basically 2 extra phosphates attached to a nucleotide, so now it has 3 phosphates. When these phosphates break, it releases energy that is used to attach it to the new strand of DNA. The DNA polymerase 3 then finishes the process, going to the end of the chain, forming the leading strand. However, half the strand is not yet created. Now, the same process occurs, but the pair of enzymes jump to the other side of the DNA and continue until it reaches the origin of replication. However, it does not do this in a continuous motion like the leading strand. Instead, it is separated into segments called Okazaki fragments. Another enzyme, DNA polymerase 1, travels back 3' carbon to 5' carbon, changing the uracil to thymine, and checking for any other mistakes. However, in between the lagging and leading strand and the Okazaki fragment, there's a gap where there's no phosphodiester bond. This is where a pair of ligase comes in and creates the bonds. Also, at the ends of each chromosome are telomeres. They're a repeating pattern of DNA that doesn't code for anything. They're there as a buffer zone because each time the nucleotides of a primase are cut out in the lagging strand, it cannot be replaced. This eventually makes the DNA shorter and shorter. This is why cells can only divide a set number of times. After this is completed, so that each nucleotide is duplicated in each chromosome, the cell can go on with the rest of the cell cycle. The The sole purpose of DNA is to code for proteins. The first step of protein synthesis is transcription. This is usually sparked by a signaling molecule that will wiggle into the nucleus and activate a promoter protein. This protein controls where a specific gene starts and when it will be expressed. The signaling molecule will bind to this protein, activating it. Then, an enzyme, RNA polymerase, comes by and binds to the promoter protein. This is when transcription starts. The RNA polymerase is the only enzyme needed for transcription. It goes through the same process of DNA duplication, like breaking and forming the hydrogen bonds, and matching nucleotides all on its own. The RNA polymerase creates a strand of messenger RNA, or mRNA, by pairing the nucleotides. However, unlike DNA replication, instead of T matching with A, RNA matches A with uracil, U. As it moves down the strand of DNA, it eventually gets to a terminator protein. This tells the RNA polymerase to stop, and the mRNA detaches. The mRNA then wiggles out of the nuclear pores into the cytoplasm of the cell. Here it finds ribosomes, where translation occurs. Proteins are made of a chain of amino acids, and to code for each individual amino acid, the mRNA is separated into codons. A codon is made up of 3 nucleotides. Each codon has an amino acid it codes for. Scientists have found out what amino acid each codon codes for, and it shows in this graph. To form the polypeptide chain, transfer RNA or tRNA carry an anticodon and an amino acid. The anticodon is the corresponding nucleotides of the codon in mRNA. After the ribosome have matched each codon with its amino acids, the polypeptide chain goes on to finish its journey to become a protein and other organelles in the cell. Mutations occur when the cell is damaged or when a mistake happens during DNA replication. When a gene mutates, the nucleotides that it consists of changes. There are 2 types of mutations: point mutations and frameshift mutations. Point mutations are when a nucleotide changes from one to another, while frameshift mutations delete or add a nucleotide. A point mutation can have no effect on a protein because it will code for the same amino acid. However, if it codes for a different amino acid, it will become a new protein. This is called missense. An example is sickle cell disease. It is caused by a single nucleotide changing and causes the hemoglobin to become dysfunctional. Another type is nonsense, when a nucleotide mutates and codes for a stop amino acid, terminating the process and creating an incomplete polypeptide chain. The other group of mutation is frameshift. When a nucleotide is added or deleted, every single codon after it will be different. This can drastically change the protein the DNA codes for. An example of mutation in our everyday lives is cancer. It is caused when a mutation causes cells to duplicate unchecked. This can happen in the lungs of the smoker because the chemicals can damage the cell and physically break or change the DNA. Cancer cells do not stop duplicating, and it will eventually kill the individual because it absorbs all the nutrients that the vital organs need. All these mutations can be better or worse for the organism, and natural selection will allow the good traits to stay and kill all individuals with harmful traits. So how does all this account for the glowing bacteria in the pea-glow lab? First of all life contains DNA and all the nucleotides that codes for proteins that will eventually allow the bacteria to glow. The pea-glow bacteria was exposed to plasmids, which are just bundles of DNA. The pea-glow absorbed them and incorporated it into its own DNA. Then when we put them into the agar dishes, we provide them with nutrients so they can have the right materials for growth. In order to grow, the cells need to duplicate, and so does the DNA. The pea-glow goes through the same steps of the aforementioned process of DNA replication. However, prokaryotic DNA is in a circle. This means that the bacteria doesn't need telomeres and any spot can be the origin of replication. The fact that bacteria have DNA in a circle means that they don't have telomeres nor a certain number of times it can divide. The pea-glow uses the same process and same enzymes: helicase, topoisomerase, RNA primase, DNA polymerases, and ligase. When the DNA finishes replication, the cells divide and become 2 daughter cells of the parent. In this time, the plasmid DNA also replicates and now has created 2 bacteria with the plasmid DNA. The plasmid DNA then goes through transcription and the gene is on its way to be expressed. To start transcription, arabinose is needed to activate the promoter protein, RSE. Then, like most other life, RNA polymerase binds to the promoter and creates an mRNA strand all on its own. The mRNA then travels to the cytoplasm of the cell, where it finds ribosomes and translation occurs. The polypeptide chain is created and will be twisted and folded to be a functional protein. This protein is called GFP, or green fluorescent protein. Then after a few days, colonies of bacteria are visible and can be tested if they are expressed in the GFP protein. The introduction of the plasmid could be considered a frameshift mutation with no effect because it added a long strand of DNA to the nucleoid of the bacteria, but each codon was the same. This is because the number of nucleotides in the plasmid was divisible by 3, and each codon being 3 nucleotides long means that each codon will be the same after. The mutation is visible, and we can see that the ones without the plasmid did not glow, but the ones with the plasmid did.